Alright, hi everybody, my name is Jane, and I am a co-op at RBC right now, doing research in natural language processing, and also a researcher on a team of computer scientists at the Toronto General Hospital, doing research in brain cancer diagnosis. So with relation to that, Amir sent me this paper, so I'd just like to thank him for hosting and coordinating this event, as well as educating me on further research being done in the area. And today I'm excited to talk to you about the large-scale unsupervised deep representation learning for brain structure. And so this paper was published by researchers at the University of Southern California. And essentially their goal was to replace a software that is currently being used for brain imaging. At the moment, MRI scans being taken from various perspectives of the brain are being aggregated in a software called FreeSurfer, where FreeSurfer essentially redevelops the image of the brain in a 3D kind of model. And the goal here was to reduce both space usage and training intensity of that process and reduce it to a simple encoder and decoder, and see how effectively a machine learning approach through convolutional autoencoders could replace a software like FreeSurfer. So just moving on to a little bit of how the research was done. In terms of data, they were working with 15,000 images sourced from publicly available repositories of research on different brain ailments and malformations. So these are things like attention deficit, Alzheimer's disease, mild cognitive impairment, and these are all available online and ready for the request of public research. And so for the purpose of their study, the group had to amass data from about 9 sources. So taking that, they were receiving scans of brain tissue, of brain images, in the form of SBMRIs, so that's structural brain MRIs, and looking at images of 256 by 256 by 256. So essentially like a 3D representation of the brain. Through a set of procedures for data augmentation, through rotating images around axes, translation and such, they were working with a total of 1.1 million data points, and then took the FreeSurfer software and kind of decided to reduce the dimensionality of their data and reduce the space usage for this process. So taking away all the content around the brain, essentially any kind of internal skull matter, skin, whatever was visible in the scan, they reduced the image to the central 200 by 200 by 200, which just contained the brain. And then further, through downsampling, reduced it to 100 by 100 by 100 images. And so these were like small 3D representations of the brain. Again, as said, 1.1 million data points. Yes, Amir.
I already have a question. So you said they started from 15,000, but they augmented it to 1.1 million?
Yes.
And what did they use for augmentation?
They used rotation about axes. The paper outlines the fact that they rotated 10 degrees, 5 degrees, -5, -10, and then also did various translation procedures to do so.
On all axes?
On, yes, all 3 axes. All right. So now the main machine learning component to what this data was used for is for the purpose of convolutional autoencoders. And so how these work is that they take a input, so in this case 100 by 100 by 100, feed it into the encoder and develop it into basically a set of features represented by a 1-dimensional by 50 dimensions vector. So this vector encodes the features of the brain that would be relevant for diagnoses of various ailments, and in this case would be things like, would include features like mass, density, and just volume of certain areas. The goal here was essentially to minimize loss between the encoding and decoding of the image, and as opposed to regular training procedures which often work to increase accuracy of a certain prediction. So here, as we're using unsupervised learning, it was just a question of comparing the initial input to the final output. They were estimating loss through a mean squared error measurement, and then looking at how well the image compared to its initial input. So techniques used in the encoder for the purpose of reducing the image down to the vector, were max pooling and then unpooling for the decoder, essentially a linear activation function, and a batch normalization. The last 2 points being important for the purpose of speeding up the training of the algorithm, as more regularized data allows for faster training. Something very interesting about the way this project was structured is that they didn't just develop one model or one network for the purpose of their training, they developed 3. So I'm going to walk you through the 3 different versions of the network they developed for the encoder. And so the first one being the staged, the second one being the joint encoder, and the third one being the 3D encoder. So bear with me with the arrows. The staged encoder works in 2 parts. So you have the first stage and the second stage. In the first stage, you're taking your 100 by 100 image, so they're, as opposed to having the 100 slides all at once, they take a 2-dimensional piece of it, and then pass it in and encode it into a 1 by 50 dimension vector. They do this 100 times, so once for each one of the 100 dimensions that were remaining from the 3D original image, and then group that into 100 by 50. So this is the intermediary phase, which they then use for stage 2. In stage 2, they took the 100 by 100 image, again, encoded it to a 50 by 1 image 100 times, each individual slice that is, and then continued by passing it into the network as a 100 by 50, and further reducing it to a 50 by 1. So the goal of the second stage is to take the 100 by 100s, the full cube, and reduce it to 50 by 1. The goal of the first stage was to take the 100 by 100 and reduce it to 100 by 50. Now, what they were doing in the second stage is using that 150 by 1, testing the decoder and basically calibrating all the parameters within the decoder by then taking this process backwards and through unpooling and deconvolution, returning it to the 100 by 50. And in order to estimate loss and provide feedback and training to the algorithm, they would compare the input 100 by 50 to the stage 2 convolved and devolved 100 by 50. So this was the intermediary phase at which they did the estimation and loss. Yes?
Yeah, question. So how did they get the very first 100 by 100? Because I thought it was 100 by 100 by 100 that they downsampled to.
Yes. So they have 100, should I repeat the question? Was that loud enough in this room?
Yeah, it would be good if you repeat.
All right, so to repeat the question is, how do they get that original 100 by 100 if our input was a 3D 100 by 100? So essentially, they are taking slices of that 3D 100 by 100, just a 2D representation of it, and repeating this process 1 time on each one of these 100 slices.
Okay, so there are 150 things at the very end that come out.
100, yes.
Got it.
Yeah, so at the end you have 50 of these vectors, and 100 of them make up that one intermediary phase.
Gotcha.
So
Yes, I was cutting here.
is it the opposite? Is it decon or is it upsample? The decoder.
The decoder?
Yeah.
So the decoder is deconvolution.
Deconvolution.
Deconvolution and like unpooling and not upsampling though.
Okay.
Yes.
That's
You have a
the
question?
point, like initially the image or video is 250 by 250 by 250, right?
Yes.
And how you like squeeze to 100 by 100 by 100?
Okay, so the question being the original 255, 56, 256, 256, how does it go to 100?
Yeah.
So the 100 dimensions come from removing the excess image data around the brain itself.
Is it using a machine learning model or?
It's using FreeSurfer, the original software.
Okay.
So FreeSurfer
I see
has.
you mentioned at the beginning.
Yes.
Okay.
Yeah. So FreeSurfer has the capacity to recreate the brain structure, but also additional features
That's
like.
a commercial one?
Yes.
Oh, okay.
Did the authors give any intuition behind why they use the staged approach? Like, what was the intention here with using this over just using a normal encoder?
I believe, the purpose of using a staged approach, the way that they go through multiple approaches throughout the paper. So essentially what they wanted to do with the staged approach is to see if the intermediary values from the encoder and the decoder, when trained individually, had an impact on the final model. They later do go through the entire encoding just in one step, and then use different techniques for the third iteration of the convolutional autoencoder. But in this case here, it was just a matter of sampling which performed better. Yes?
Just to make sure that I'm understanding correctly, so the left side would be trained on 100 times the data as the right side. Like, that would be effectively like 120 million. Because each section is treated separately.
Each section is treated separately, but the thing is, for stage 2, you take the same sections taken separately. That same 100 that you had for the block gets subdivided into 100 again. So you're still using 100 2D slices for the stage 2 up until this point here.
So you do it once in the left section to like get the parameters or train the parameters or fix the parameters, and
Yes.
then do it on the right. So is that correct? Is that
Yes.
the way they do it?
Yeah. So they first train stage 1, and then using their results from train stage 1, train stage 2 to develop these vectors of 50 by 1. Yeah. Further questions? Okay, so this.
Actually, I do.
Yes.
It might be a question for you or anyone else who knows the convolution autoencoder. Like, I'm not really sure about how this works, but deconvolution is usually a very noisy process. Is that a problem here? Like, does that add extra noise when you're trying to reconstruct?
But she said they use upsampling.
No, she said
They don't use upsampling. They use
everything
deconvolution.
except that.
Yeah.
Oh yeah, because deconv adds noise, right?
Right. So exactly.
So I think that's probably the loss. Sorry.
No, go ahead.
Yeah, I think the loss function is designed to kind of mitigate that or reduce that noise.
There are a couple of papers which just discuss using deconvolution versus upsampling, and generally they show that upsampling is better.
Okay.
But
Really?
those are just experimental results. There's no actual math in there. It's just that we tried the experiment, and they showed in most cases upsampling produced less artifacts when they were going backwards.
Wow. Upsampling, you're just taking one pixel
Yeah,
and
it's
you're blowing
really
it up,
good.
right?
Yeah.
Can you like speak up?
Yeah.
Oh yeah, sometimes if you don't get the strides right when you're in deconvolution, you can have checkerboarding artifacts because
Yeah.
the strides can kind of like
Overlap.
start overlapping over each other.
So upsampling is
So I've
better.
heard.
Well, some people argue that they have experimental results to show it, but
Wow. Okay.
I think you
Yes.
mentioned this, but the ground truth for stage 2 is the output of the trained stage 1. Is that correct?
Yes.
Okay.
The ground truth for both stages is the center intermediate phase of 100 by 50. All right, so that is the stage model they trained. The second iteration for their CAE was the joint model. So the joint model is where they go straight through, and instead of training the two separately, just train it all at once. And so here you're going from this 100 by 100 slice and repeating that encoding 100 times, developing this 100 by 50 again, and then without any separate training, going through and encoding this down to a single 100 by 50. So a certain discussion that we were having is that, like, what's the difference between encoding this 100 by 50 through the process of convolution, as opposed to taking the average of all these 100 by 50 vectors. And so I guess the consensus this discussion had reached was that the average pooling would be less accurate and generate more noise data, as opposed to the convolution of all features, which kind of retains relevance and weighting. If anybody else has any other thoughts on that, that'd be very exciting to hear. So, any questions about the joint? Yes.
So how does like the back propagation chain work? Like you have to fan out from the 100 by 50 back to like 50 versions of the single frame, like single slice encoders. Is
Yes.
that correct? Okay.
Yeah, so just a question about how the back feed works. It's all about decoding this entire chain back to the 100 by 100 by 100. So there isn't an intermediary. This gets decoded up until the 100 by 50, split up, and then decoded again. And then rejoined at the end. Yes? Okay. So then the 3D method was separate from the staged and the joint, in that essentially they just used 3 convolutions on the entire piece of data for each iteration, and just convolved it to a single 1/50. So this one, even though it's 3D convolution being the simplest of them all. And then, any questions on this? Okay, so, these images here illustrate the various reproductions of the brain. And so these being the original brain images, you can see how the images produced by the different encoders and then decoders are rather very similar, but in themselves all vary by contrast to the original in the slightest bit. But overall, to a pathologist, for example, or someone working with these scans, would provide essentially the same sort of information, and as the paper commented, were very accurate reconstructions to a point where physicians couldn't tell the difference between the encoding versus the original. So, just a bit of qualitative analysis. Oh, a question here?
Sorry, the deconvolution process for the 3D con, did
Yes.
they talk anything about how they did it? Because usually the decon layers are for 2D cons.
They did not discuss in detail as to how they did the deconvolution, but the understanding was that using the kind of convolution voxels, equivalent to a kernel in 2D, they did the same thing in 3D, so
3D.
deconvolution voxels.
They're probably doing their own stuff.
Yeah.
Well, what is your concern there? Like,
No, I was just curious because I've never seen this.
Just in terms of implementation.
Yeah, in terms of implementation.
All right. So qualitatively speaking, what they did next was essentially generate mappings of the effects of different voxels on the overall brain image scan. So what you're looking at here are saliency maps created through Keras-Viz, where you see the importance / value that each voxel creates to the overall representation of this image. So,
Sorry, quick question.
Yes.
What's a voxel?
A voxel is a 3D kernel.
Okay.
Yeah, so the same way you would use a 2D kernel for convolution, just the term they use. Yeah.
Cool.
Is it a kernel or like a pixel in 3D?
It's equivalent to a
volume
kernel.
pixel,
It's
yeah.
equivalent to a kernel.
Oh, is it? Okay.
Yeah, not a pixel. It's just like the kernel being the numerical like slider that goes across. That's the value of the voxel. The voxel has 3D. It's like a 3D matrix illustrating all the like the weightings for a kernel. Whereas a pixel is the values that are encoded in this location.
Actually, for point clouds, they use the term voxel for.
Yeah, I think it's
pixel.
like a volume
it's pretty
pixel.
common to
Yeah,
be
it's
like
pretty
a
common.
pixel.
Yeah.
It's just a term.
It's just
Okay.
a term, I think.
I guess. All right. So in these saliency maps, they represent all the value of the perturbations of the voxels to each one of the brain scan images. And so here, broken down to separate embeddings. Another thing they noted is the fact that each one has different values, like different significances for different features, and ensuing that these images, though they're going through a similar convolution and deconvolution process through the encoding and decoding, all map the importance of different features differently. So I guess that was what was relevant to pick up from these maps. Any questions for these images?
How did they determine saliency? Like, how did
Saliency, basically, what they defined it as was the value for the voxel. So the higher value of voxels, like once convolved, producing greater brightness for the
got
pixel,
it.
illustrate where you see the brighter spots in the map. Yes?
So these are like feature map values, not like
Yes.
gradients with respect to like different voxels in the input.
Yes.
Okay.
Yeah, these are feature map values.
Which is basically the receptive fields of the pixels in the voxels. So
I'm
from
not
a
sure.
kernel, from 3D kernel follow along the feed forward, say you have some activation.
Mhm.
That activation can be linked to pixels in the original image.
Yes.
that's-
These are mapping for the activation of each individual
Yes, basically the
pixel.
receptive
Yes.
fields of the for the features later on.
Yes.
Yeah.
Further questions?
One other question.
Yes.
They actually kind of try to map these or align these with like the neuroanatomy of like the brain. Are they like regions that are maybe implicated in like Alzheimer's or like other like diseases or anything like did they actually do any sort of analysis on that or?
So the question was, are they taking these images and seeing if they map back to any illustrations of neurodegenerative disorders? So essentially, are these relevant to the diagnosis of, let's say, Alzheimer's or mild cognitive impairment? And no, the paper does not discuss the fact that they mapped it. What they were just mostly giving attention to was the fact that these are being mapped differently amongst the different presentations. So there's no way from these ones here to analyze these maps here to analyze exactly which features are relevant for the diagnosis. These were just mappings of how the kernel interacts with the rest of the input. Yes?
On that topic, I read another paper which did something very similar, except they used the reconstruction loss to do anomaly detection, and then they didn't actually diagnose anything, but they just showed that things which follow the pattern of a healthy brain versus things which are considered anomalous. Doing something very similar, but not exactly this.
Very interesting. I guess maybe Amir could share that paper if linked to it.
Or you can share it on Slack yourself.
Yes. All right. So then further, they did. Oh yes, Amir.
Remind me why there are 10 images. Like what are?
They essentially. So the reason there are 10 images is because they had to take embeddings of the 3D representation. So these ones being the embedding for like a single node, and they essentially map 2 nodes, so 2 locations in the brain. And this breaks down essentially the depth of one brain scan. So if you were to put all these 10 together, you would be able to form like the right half and the left half of the brain.
I see.
Yeah.
Good question.
Yes.
So, like, were these images taken from like the same MRI, or were they from different MRIs? We don't really know. And like, does it vary? I'm just wondering if it varies from machine to machine, like quality of image and other different, quality of image and things like that.
So the question was same MRI or different MRIs in terms of where these images are taken, and more specifically, these were produced like by the researchers, these saliency maps. So these aren't MRI images, but the ones that they were taking were coming from a number of data sources. So you would get some variation in quality and variation in, I guess, a lot of some of the dimensional parameters in terms of how the images were taken. In a sense, I guess that's advantageous for the sake of data, for the lack of overfitting and more data variation. Yeah. Thank you. All right. Any more questions on this slide? All right, so moving forward to the quantitative analysis. So here what they essentially analyzed was the ability for these images to be used for, it keeps punching on me, it scares me. So they essentially took these images and analyzed them for the capacity to be used for diagnosis. So the first column here illustrates the accuracy score of a FreeSurfer generated structural brain MRI image and compares it to that for the staged, joint, and 3D representations of the brain. So what they were using is essentially different, they're using random forest models and linear regression for the purpose of diagnosis in this case, and being able to classify the results, like the encoding outputs, under a certain category for disorders in the brain. And here they tested it for things like Alzheimer's versus attention disorder, attention disorder for cognitive impairment, and so forth. And overall, the stage 3D, the 3D version of the encoder tended to perform better. And so there were kind of the impact of this research isn't necessarily in the increased performance of the models, but more so in the time that it took for each one of these diagnoses and these images to be generated. So FreeSurfer takes about 2,047 hours to generate one structural brain MRI image, whereas the encoders took 0.5 seconds, 0.45 seconds on average to generate these same images. So one consideration here, maybe this will address a question, but was the fact that they weren't necessarily optimizing for training accuracy. Because what they're doing here is testing the reconstructibility of images and comparing the images to that of FreeSurfer. They were using the classifier trained separately from this entire algorithm, being random forest and logistic regression, to not essentially making the goal of this paper not diagnosis, but reconstruction of images. So had they been optimizing for image, for classification and diagnosis, they could have included it in the networks for convolution and then further training in that sense. But their optimization was comparing the input to the output, not comparing the classification of the output. Yeah, so questions?
So my question was, don't they use the FreeSurfer input as input to this model? So that input they initially had, does that also take 2,000 hours to make?
The original input, so the question is the input coming from FreeSurfer? And in part, but the original input is actually just the image of the MRI scan, and they're using FreeSurfer in order to remove the excess information in the image. So they remove the skull with FreeSurfer, but they don't actually generate the full structural representation of the image with FreeSurfer. The structure is represented through the convolutional autoencoders. We had a question over here?
Yeah, I'm just curious. I don't know if you know the answer, but like, if it takes 2,047 hours for each image, like, how do they even do that in practice? Like, do they just run on like 100 cores or something and parallelize over a day?
I
like,
can
yeah,
safely say that I don't know how this is
that's fair. It
at
seems
all
like,
in practice, but yeah. If there are any FreeSurfer experts in the room, that would be a cool thing to address.
24 by 7.
100 days.
$2,000, please.
Yeah.
100 days.
A lot of time.
15 months.
We had more questions in this area?
I had kind of a similar question, because it seems to me that like the background removal part that they actually share with like the FreeSurfer like front end would be the part that is more kind of combinatorial, whereas like the hand-coded features don't seem to be the part that should take like 2,000 hours. Yeah, which is kind of, they don't really discuss the this part, like how they actually configured this, or if anyone actually uses this as like as a tool in like an actual like practice, or they don't really go into any detail regarding this, the 2,047 hours, because that's really odd and like pretty close to like 2 to the 11. So
that's for one image.
Yeah,
for
so
one image, yeah.
unfortunately, no, they don't go into too much of the detail around FreeSurfer. I guess based on this paper, it is what it is. And that's like the main reason for optimizing this whole procedure. I guess that's the motivation behind.
Even if it's 20 hours, it's a huge optimization. So.
Yeah, very true.
But like the pre-processing was just like essentially clipping the images, right?
Yeah. The
So
pre-processing
I don't think
is.
that takes a lot of work. Because
Yeah.
you can probably write simple rules to do that.
I don't know why they use FreeSurfer for that though.
I mean, it is built in. They might as
It's
well
always
use it.
easier
so-
to do it on.
Yeah. I don't know what other software exists out there for the purpose of removing skulls from around the brain. I guess it's specialized industry work, so they go with that. We had a question here?
Yeah, I was going to ask what makes a structural MRI different than our other MRI for that purpose.
So difference between a structural brain MRI as opposed to a regular MRI. Regular MRI is 2D. Here they're using 2D images to reconstruct a structure of a 3D image, and analyzing things including density, depth, volume, something that you would not be able to capture with a 2D image of a brain MRI. Yes.
We just given one example of regular MRI and SMRI.
Point noted.
So
yes.
So what are, I didn't quite get what each of the rows are, because there are two malfunctions, I guess, for each of the rows, like AD and MCI.
Yes.
Is that the ratio of their accuracies, or?
That's a comparison. Yeah, these are conditions, and this is the diagnosis, like the differentiation and classification between two conditions. So when fed only images of attention deficit versus mild cognitive impairment, it was able to classify with a 71% accuracy as opposed to a 72% accuracy.
So it's a binary classification between those two for each of the rows?
Yes, binary classification between two
Okay.
different kind of
I see.
images. And we had another question here, no? Russell? No? Okay. All right, and so just in conclusion, while this method is not necessarily optimizing for reconstruction classification accuracy, they do kind of like a mile's work in terms of efficiency, time taken to reconstruct images, and then volume of data used and essentially processing power, optimizing the process for regenerating structural brain MRIs for the industry. Any further questions on the rest of the paper? Yes?
I wonder that you mentioned about at the beginning they are starting with 15,000 image, am I right? Or
Yes.
yeah, something like that, and then they tweaking it to the 1 million, I guess.
Yes.
And then one of the approaches is the 3D one.
Yes.
You mentioned that they are basically rotating it 5 degrees each time.
Mhm.
So I wonder that if I'm going to use a 3D, why I need that rotation?
I guess the rotation is most advantageous. So the question was, if we're using 3D convolution, what's the point of generating that rotation? And so I would probably have to, like, it's not discussed in the paper formally, but I would assume that the rotation is most valuable for the purpose of the staged encoders. As you noted, it's not necessarily most relevant for the 3D convolution, but for the purpose of generating data and kind of augmenting their set, it was probably helpful for staged and joint.
The other question regarding of the two-stage one that you mentioned.
Yes.
Basically, they're calculating the loss function separately from in the each stage, am I right? Or?
Yes, loss functions. So the loss function between the two stages is calculated separately, and then the original kind of encoding and decoding are compared at the center intermediate 100 by 50 stage.